Picture this. You've got a container of leftovers sitting on your kitchen counter.
We have all been there.
Right. You meant to put it in the fridge after dinner, but maybe you got distracted by a phone call or, like, a movie, and now it's just been sitting out at room temperature overnight.
Which is obviously a recipe for disaster.
Exactly. Because we all intuitively understand what's happening in that container. Bacteria are multiplying. It's the whole reason you're going to end up throwing that food in the trash. But the actual mechanical process of how those microscopic organisms divide, how one single cell becomes millions while you sleep, is, well, it's infinitely weirder and way more highly choreographed than what we were taught in high school biology.
Oh, totally. And a lot more controversial, too. I mean, it fundamentally challenges our basic assumptions about life. We are so conditioned to think of bacterial division as this simple biological copy-paste function.
Just little factories stamping out clones.
Right. But when you examine the molecular mechanics, it is a masterclass in cellular engineering. It comes complete with physical aging and really complex geometrical problem solving.
And that is exactly our mission for today's deep dive. We are unpacking binary fission, which is the primary mechanism by which bacteria divide and reproduce.
And we have some incredible source material for this one.
We really do. We're drawing from this fascinating stack of historical letters, evolutionary data sets spanning over 70,000 generations, and some groundbreaking modern studies to figure out how this unseen world actually operates.
Though, to really appreciate the complexity, we should probably start by defining what we think binary fission is, right? Just before we complicate it.
Yeah, let's set the stage with the classic textbook definition. So if you look up binary fission, the standard definition tells you that a single prokaryotic cell replicates its genetic material and then splits into two equal identical daughter cells.
Very neat and tidy.
Right. It's asexual reproduction. There is no fertilization, no combining of genes from two parents, just one cell splitting perfectly in the middle to become two identical clones. It sounds clean, simple, and very easy to memorize for a test.
But that clean, simple definition is essentially a lie of omission.
A lie of omission.
Yeah, I mean, it hides about 2 centuries of scientific complication. Because when you peer through a modern microscope, that textbook definition just shatters. You find that this process involves a measurable form of physical aging. It involves molecular machinery that acts like microscopic scaffolding.
It's not just a balloon popping in half.
Not at all. And there are evolutionary mysteries in there that biologists are still actively debating today.
So if a bacterium is just, a bag of cytoplasm splitting perfectly in half, why wouldn't the two resulting cells be perfect clones?
Well, first, we have to discard the myth of genetic perfection. The enzyme responsible for copying the cell's DNA is called DNA polymerase. And while it is incredibly efficient, it makes typos.
Like transcription errors.
Exactly. Now, it is a very low error rate. In a standard bacterium like E. coli, it's roughly 1 mistake for every billion base pairs it copies.
Okay, that's pretty good.
It's amazing. But that non-zero rate means the resulting daughter cells are practically never strictly identical. They are just almost genetically identical.
Okay, I can accept a few genetic typos. I mean, nothing in nature is mathematically perfect.
Yeah.
But I want to push back on the idea of physical differences. How can two halves of the exact same cell not be physically the same?
This is where the biology gets wildly counterintuitive. They aren't physically equivalent because of how the cell wall is constructed. There was this landmark study published in PLOS Biology back in 2005. A team of researchers decided to track individual E. coli cells under a microscope across multiple generations.
Just watching them divide over and over?
Right. And what they documented was a phenomenon that revolves around the old pole and the new pole of the bacterial cell.
Okay, let's unpack this. When a rod-shaped bacterium like E. coli divides right down the middle, the two new daughter cells each get one of the original older ends of the parent cell, right?
Exact.
And that older end is the old pole. And then they each get one freshly created end from where the physical split just happened. So that's the new pole.
You've got it. Every single division creates this asymmetry. And what that 2005 study revealed is that the daughter cell inheriting the older end of the cell wall, the old pole, actually grows at a slightly slower rate.
Wait, really?
Yeah. And it's marginally more likely to die than the daughter cell that inherits the brand new pole.
Oh, wow. It makes me think of, inheriting a car. Imagine one sibling inherits a vintage car and the other gets a brand new model straight off the lot.
Okay, I like where this is going.
The vintage car, which is the old pole, has simply been on the road longer. It requires a bit more maintenance. The parts are a little more worn down, so it's slightly more likely to stall out compared to the brand new model.
That is a brilliant analogy, and the scientific term for that biological wear and tear is cellular senescence.
It's aging.
Basically, yes. Before this data came out, the working assumption in microbiology, an assumption that had literally stood unchallenged since the 19th century, was that binary fission was a form of reproduction completely free of aging.
People just thought they were immortal.
Yeah, biologists assumed bacteria just divided and stayed eternally young forever. But this measurable asymmetry in cellular senescence proved that bacteria do, in a very real way, age.
Which fundamentally changes how I view that puddle of spilled milk on the counter. But it leads me to an even bigger mechanical question.
Let's hear it.
Knowing that division leaves one cell slightly older and one newer, how does a single cell, which has no brain, no nervous system, and no tiny ruler to measure itself, actually execute this physical split directly in the exact middle every single time?
To understand how a cell finds its center, we have to break down the 4 sequential jobs the cell must complete. It is a highly coordinated, 4-step choreography.
Okay, step 1.
Step 1 is replicating the chromosome. Unlike human cells, which store DNA in a nucleus and start copying from dozens of different spots all at once, most bacteria have a single circular chromosome. And it only has one starting point, which biologists call the origin of replication, or oryx.
I always pictured it like unzipping a jacket, starting from the top and going down.
It's actually more like unzipping a jacket in both directions at once.
Oh, weird.
Yeah, 2 replication forks fire simultaneously in opposite directions from oryx and travel all the way around the circular DNA until they meet at the far side. They meet at a specific sequence called the terminus, or ter. It's highly efficient.
Okay, so the DNA is copied.
Right. But then comes step 2, which is segregation. You now have 2 complete copies of the chromosome tangled up in a remarkably tiny space, and they have to move to opposite ends of the cell before the split happens.
Now, for a long time, the scientific consensus was that they just sort of drifted apart passively, right? Like just floating away from each other as the cell grew.
Yeah, that was the prevailing theory for decades. But it turns out it is not passive diffusion at all. The cell uses specialized partitioning proteins, specifically 2 proteins called para and parb.
And what do they do?
They actively push and pull the chromosomes apart. Parb binds directly to the DNA near the origin, and para forms a sort of gradient, polymerizing and acting almost like a microscopic game of tug-of-war.
So they're literally dragging it.
Physically dragging the DNA copies to opposite poles of the cell. It's a very deliberate, energy-consuming, mechanical sorting process. Which brings us to step 3, building the divisome.
The divisome being the actual division machine, right? And this involves a specific protein called FTSC, which seems to be the hero of this entire biological process.
Unquestionably. The discovery of FTZ by Aaron Lutkenhaus in 1991 is arguably the most important breakthrough in modern binary fission research.
It's a catchy name, too.
Well, the name itself is actually an acronym for Filamenting Temperature-Sensitive Mutant Z.
Rolls right off the tongue.
Right. Researchers found it by studying mutant bacteria that couldn't divide. Instead of splitting, these mutants just grew into long, continuous filaments. But what normal FTSC proteins do is polymerize to form a contractile ring, known as the Z ring, at the exact future midpoint of the cell.
See, wait, this is where I get tripped up, because this sounds like highly coordinated factory work. How does a microscopic ring of proteins know where the exact middle of a rod-shaped cell is?
What's fascinating here is that the cell solves this engineering problem using first principles of geometry. It relies on 2 competing regulatory systems. The first is called the MIN system.
Like MIN.
Yes, the name comes from mutant bacteria that produce miniature cells because they divide in the wrong place. In a normal cell, you have 3 proteins, MIN-CU, MIND, and MINE, and they literally oscillate.
Oscillate, like a fan.
It's like a metronome. They bounce back and forth from one pole of the cell to the other. Because they spend the vast majority of their time at the extreme ends of the cell and very little time in the middle, they create a chemical zone in the exact physical center where the Z-ring is allowed to safely assemble.
Oh. So it's a process of elimination. It's like trying to find the exact center of a pitch-black hallway while blindfolded, but you have these bouncy bumpers set up like in a bowling alley, constantly bouncing you away from the gutters.
I love that.
So the MIN proteins act like those bowling bumpers, pushing the FTSC proteins away from the poles so they have no choice but to settle perfectly in the center.
That captures the physics of it beautifully. But the cell doesn't just rely on the bumpers. It has a second fail-safe system called nucleoid occlusion.
Which sounds intense.
It's a mechanism that physically prevents the Z ring from forming directly on top of the unsegregated DNA. A specific protein coats the DNA and blocks FTSD. If the ring formed over the DNA, it would essentially guillotine the chromosome as it closed.
Yikes, that would be bad.
Very bad. So, between the Min system bouncing the ring away from the ends and nucleoid occlusion blocking the ring from strangling the DNA, the cell finds its midpoint perfectly. Then finally, we reach step 4, constriction.
Right, and this is where the Z ring acts like a biological purse string, pulling the cell membrane shut, while a dozen other proteins work frantically to synthesize the new cell wall, which is called the peptidoglycan panseptum, building it inward until the two halves finally detach.
Precisely. It is mechanical perfection on a nanometer scale.
Okay, but all of this mechanical perfection sounds like it would take hours to coordinate. I mean, the DNA tug-of-war, the oscillating proteins finding the middle, the purse string closing. But we constantly hear about how insanely fast bacteria multiply. What is the actual biological speed limit on this cellular factory?
Well, the speed limit varies wildly depending on the specific species of bacteria and its environment. But the absolute speed champion of the microbial world is a marine bacterium called Vibrio nitrogens.
Where does that live?
It was first isolated from the mud of a Georgia salt marsh back in 1958. It can complete this entire 4-step division cycle from DNA replication to physical splitting in under 10 minutes.
Wait, under 10 minutes to build an entirely new cell from scratch?
Yes. For decades, it was largely ignored in the scientific literature. But in recent years, synthetic biologists have rediscovered it. Because of its incredible metabolic rate, it has become a darling of the biotech industry as a super-fast alternative to traditional lab bacteria for generating proteins and running experiments.
Since you mentioned traditional lab bacteria, that brings up E. coli, which is the standard everyone thinks of. The rule of thumb for E. coli is 20 minutes, correct?
Under ideal laboratory conditions, yes. If you put it in a rich nutrient broth and keep it at exactly 37 degrees Celsius, it divides roughly every 20 minutes. There's a classic thought experiment that illustrates the sheer terror of this math.
Let's hear it.
If a single E. coli cell divided completely unchecked every 20 minutes for exactly 24 hours, it would undergo 72 divisions. That gives you a mathematical potential of 2 to the 72nd power cells.
Which would probably produce a biomass heavier than the Earth itself.
Exactly.
But obviously, we aren't buried under a mile of E. coli.
Thankfully, no, because that log phase exponential growth is eventually halted by physical reality.
Right.
The bacteria run out of consumable nutrients, their own metabolic waste accumulates and poisons their environment, and they literally run out of physical space to expand. But it perfectly illustrates why bacterial growth curves shoot straight up exponentially during those first few hours on a kitchen counter.
Okay, so Vibrio and E. coli are the sports cars. But on the other end of the spectrum, you have the absolute slowpokes. Our material highlights Mycobacterium tuberculosis, the pathogen responsible for TB. It takes 15 to 20 hours to complete a single division cycle.
Which represents a massive paradigm shift when you think about clinical medicine. That single mechanical fact, a 15-hour generation time, explains so much about how we treat disease.
How so?
It's the exact reason why standard treatment courses for tuberculosis run for 6 straight months, or sometimes even longer. The antibiotics we use typically target bacteria only when they are actively in the process of dividing, specifically when they are building that new cell wall.
Ah, I see.
Yeah, so if the pathogen only attempts to build a cell wall once a day, you have to maintain a high concentration of the antibiotic in the patient's body over many, many slow division cycles to ensure you eventually catch and kill the entire population.
But I'm struggling to reconcile that speed with the danger it presents. If TB divides so incredibly slowly, why is it one of the deadliest infectious pathogens in all of human history? Doesn't a faster division time mean a more dangerous infection?
This raises an important question, and it gives us a chance to dismantle a major public misconception. We are constantly conditioned by shorthand in the media to think fast equals dangerous.
Right, the flesh-eating bacteria sweeping the nation.
Exactly. But virulence, which is the scientific measure of how deadly a pathogen is, has very little to do with its division speed. Virulence is dictated by the specific toxins the bacteria produce, how well their cell walls evade the human immune system's radar, and how easily they transmit from one host to another.
So speed isn't everything.
Not at all. TB is a slow, chronic, and incredibly destructive disease exactly because it plays the long game. It has a thick, waxy cell wall that immune cells can't easily break down, allowing it to hide out in the lungs for years.
That makes a lot of sense.
Mm,
A fast-growing infection might overwhelm the body quickly, but a slow one can evade detection and cause long-term structural damage. That brings up another media shorthand we see constantly regarding antibiotic resistance. The narrative is usually bacteria multiply so fast, they evolve resistance instantly.
yes, and that narrative dangerously conflates two very different mechanisms. Fast division on its own does not create antibiotic resistance.
It doesn't.
No. Fast division simply increases the raw number of opportunities, the sheer volume lottery tickets, if you will, for a random genetic mutation to occur during DNA replication. Or it increases the chances for a resistance gene to be acquired from a neighboring bacterium.
Oh, so it's a numbers game.
Completely. If the surrounding environment happens to have antibiotics in it, that random mutation gives the cell a survival advantage and it gets selected for. But the speed itself isn't the cause of the resistance.
Right.
This oversimplification obscures the real message of antibiotic stewardship, which is about finishing your entire prescription to eliminate every last bacterium before those extremely rare random mutations ever have a chance to take over the population.
Understanding how this molecular process drives random mutations provides the perfect bridge into how humans actually figured all of this out in the first place. Because the history of studying binary fission is essentially the history of humans trying to build tools to watch evolution happen in real time.
And it is a surprisingly long history. It starts all the way back in 1676 with Antony van Leeuwenhoek.
Ah, the lens grinder.
Yep, he was a Dutch draper who ground his own glass lenses and became the first human being to ever observe bacteria, which he famously dubbed animalcules. He could see that there were more of them over time, but he had absolutely zero framework for understanding how they multiplied.
Because in the 17th century, the scientific consensus was still largely focused on spontaneous generation. The idea that rotting meat just magically produced maggots, or that dirty water just birthed microbes out of thin air.
Exactly. The concept of microbes having biological parents didn't exist yet. It wasn't until the 1860s that Louis Pasteur finally demolished the spontaneous generation theory with his famous swan neck flask experiments.
I remember learning about this.
Yeah, he boiled nutrient broth in a flask with a long S-curved neck. Airborne microbes got trapped in the curve of the glass, so the broth inside stayed completely sterile indefinitely. But the moment he broke the glass neck off, allowing dust and microbes to fall in, the broth spoiled immediately.
Which definitively proved that microbes must come from prior microbes. They don't just spontaneously generate from the broth. That experiment established the absolute baseline for understanding binary fissionism and all of it. It is true biological reproduction.
From there, you have figures like Ferdinand Cohn in the 1870s who helped classify these organisms, establishing that they had reproducible, predictable life cycles, but the actual internal molecular mechanics, the understanding of how the DNA interacts with the cell, that didn't arrive until almost a century later.
In 1963, right?
Yep, when Francois Jacob, Sidney Brenner, and Francois Cousin proposed what they called the replicon model.
Okay, I want to make sure I understand this replicon model. What exactly were they proposing back then?
Well, they were trying to solve the problem of segregation before anyone knew about those para-An, para-B tug-of-war proteins. Jacob, Brenner, and Cousin hypothesized that the bacterial chromosome didn't just float freely in the cytoplasm. They proposed it was physically anchored to the cell membrane. Their model suggested that as the cell grew and the membrane elongated, the physical stretching of the cell wall would simply drag the two copied chromosomes apart.
So the growing outer envelope of the cell does the heavy lifting of separating the internal genetics.
That was the idea. Now, today, we know the reality is much more complex, involving those active partitioning proteins we discussed earlier. But the replicon model was a massive conceptual leap because it was the first time scientists explicitly linked the process of DNA replication to the physical growth of the cell membrane. It set the stage for all modern microbiology.
Here's where it gets really interesting, though, because once we understood those basic mechanics, biologists realized they could use the sheer speed of binary fission to their advantage. I want to talk about Richard Lenski's long-term evolution experiment, often just called the LTE.
Such a cool project.
This started back in 1988 at Michigan State University. Lenski took populations of E. coli and essentially just let them divide in flasks day after day, year after year, transferring a small amount to fresh broth every single day.
And that experiment is incredibly still running today. Those bacterial lineages are well past 70,000 generations at this point.
It is like putting a time-lapse camera on an evolutionary process that normally takes millions of years for larger animals, suddenly making the invisible math of binary fission visible to the human eye.
It's one of the most important continuous data sets in evolutionary biology. Because binary fission is so fast and purely asexual, every single mutation that occurs can be tracked and traced back through the generations. The most famous milestone in the experiment came around generation 31,500.
What happened then?
After years of predictable behavior, one specific lineage of E. coli suddenly evolved a novel trait, the ability to metabolize citrate as a food source in the presence of oxygen.
Which is a metabolic ability that the ancestral strain of E. coli completely lacked.
Yeah.
It's literally evolution acting on the genetic variation generated by binary fission errors caught on tape.
And yet, even with all of this historical data and modern genetic tracking, there are still massive debates and bizarre exceptions in the field. Remember the textbook definition we started with? One cell splitting into two identical halves.
The one we already established was a lie of omission due to aging.
Right, but some bacteria defy the physical geometry entirely. Take a bacterium called Caulobacter crescentus. It purposefully divides asymmetrically.
Doesn't even try to split evenly.
Not at all. During division, it runs two distinct genetic programs in the same cell, localizing different proteins to different poles. The result is one highly mobile swarmer cell equipped with a flagellum to swim away, and one stationary stalked cell that anchors itself to a surface.
That's amazing.
Two completely different physical structures resulting from a single division. It proves that binary fission isn't just a crude mechanism for population growth. It can be a highly regulated developmental decision.
Now, you mentioned earlier that most bacteria use that FTSC protein for their division ring. But nature loves an exception. Does this exact same division machine operate across all single-celled organisms, or are there variations? I'm thinking specifically about the archaea domain.
This is where we hit a major evolutionary mindbender. Archaea are single-celled organisms that look like bacteria but are genetically distinct. And it turns out some archaea, like a genus called Sulfolobus, do not use FTSC at all.
They don't.
No, instead they use a completely different protein system called ESCRT3 to divide. That stands for endosomal sorting complexes required for transport.
So they use a totally different set of tools to achieve the same split. Why is that an evolutionary mindbender?
If we connect this to the bigger picture, it has to do with us. The FTSC protein used by most bacteria is evolutionarily related to tubulin, which is the protein that builds the structural scaffolding during cell division in complex eukaryotic cells like human cells.
Okay, I follow.
But the ESCRT3 system used by those archaea. Human cells use that exact same protein family for a different purpose entirely. We use it for remodeling our internal membranes and pinching off transport vesicles.
Wait, so both of these ancient single-cell division mechanisms are evolutionarily related to the machinery operating inside human cells right now?
Yes. You have binary fission acting as a catch-all umbrella term for at least two distinct ancient molecular toolkits, both of which have deep, surprising evolutionary ties to how our own complex cells function today.
So what does this all mean for you, the listener? Let's bring this microscopic world back down to Earth. Today, we thoroughly dismantled the illusion of the simple, identical clone. Binary fission is not a basic copy-paste job. It is an intricate, mechanical, aging-prone process involving protein metronomes and DNA tug-of-war.
It is the biological engine that dictates the fundamental pace of microbiology. The mechanics of the divisome and the geometry of the Z-ring are the underlying reasons why food spoils on your counter, why antibiotic dosing schedules demand such strict compliance, and why researchers can literally track evolution in real time in a laboratory flask.
The next time you throw out that spoiled milk, or you force yourself to take those last few days of an antibiotic prescription, even though you already feel better, I want you to realize you are interacting directly with the mathematics of the Z-ring.
You are fighting against a microscopic engineering marvel that has been ruthlessly refining its division strategy for billions of years.
It really makes you respect the leftover container a little more. And I want to leave you with a final thought to ponder based on everything we explored today. We started by breaking down the myth of the identical ageless clone. We learned that bacteria can experience senescence. They physically age based on inheriting the old pole. We also learned from Caulobacter that some bacteria can make developmental decisions to produce structurally different offspring with different specialized jobs.
Right.
So if a colony of bacteria can experience physical aging, and can diversify their cellular roles to survive. Wait, sorry. If they can diversify their roles to survive, does that mean a puddle of bacteria on your kitchen counter is actually functioning much closer to a complex, aging multicellular organism than just a random collection of identical clones? Next time you see leftovers left out too long, think about that.